I also was invited as an educational faculty to talk about new data for the field in the MET and HER2. So it's interesting. We just talked about MET exon 14, but in lung cancer, MET is no longer just MET exon 14. So I think the MET alterations start to expand, which is good for the patients because more patients could be using targeted therapy and derive benefits. So at the beginning of the talk, we will be reviewing the MET as a target, either exon 14, MET as a fusion protein, or MET as an overexpression. So in those three scenarios, we have different level of evidence from clinical trials, and then we have different approaches we can develop drugs. Probably, again, goes back to a biomarker for MET overexpression. I think the protein level detection is almost required to develop a biomarker-driven drug there. After reviewing the classification of MET alteration, next one, I briefly review the approved MET TKI, especially for MET exon 14. We're familiar about those information and knowledge, but we'll just set a bar there, basically thinking we have good standard of care, how can we improve upon? And next, I will be reviewing some of the new data coming from antibody and also coming from ADC drug, for example, Teliso-V, that has great preliminary data in the MET overexpressed lung cancer patient population. In that particular trial, genetic testing is not used to guide as a biomarker, rather than the MET immunohistochemistry is used as a biomarker as appropriate. And then in the end, we wrap up with talking about newer approach, newer drugs on the horizon. There are a number of them coming through the pipeline, including small molecule, antibody, ADC, combination approach, as well as immune approach. So I think it's really an exciting field moving forward. After we finish talking about MET, we'll also be talking about HER2. As I said, it's a new target with only one FDA-approved ADC drug. We use a similar format reviewing existing drug and review the unmet need. In my eye, I think the unmet need will be not just improving what we have, but developing small molecules so that we can serve the patient once they progressed on prior treatment. The truth is there are multiple very good HER2 small molecules in development, with some of them have promising initial efficacy data. So we'll be talking about some of those drugs as well as combination approaches for HER2. HER2, I think resistant mechanism is still not well understood because the field is still young. So some of the effort probably going to be directed towards understanding resistance to different type of drugs for HER2.